All right, good morning. Yeah. Would someone close the door, perhaps? I guess we still have another person coming by. So good morning, welcome to session 5. Let's see. We're going to skip ahead in the book a little bit to chapter 5, just because this is the next major thing that you will need to know about for your project, and it seemed to make sense in the sequence of the course. So I'm going to introduce you to definitions and ideas of distances in high dimensions and motivate why distances are so important in descriptive and exploratory high dimensional data analysis. And related to this, we'll cover principal components analysis and multidimensional scaling, also known to some people as principal coordinates analysis in other fields. So the reason we use distances is because in high dimensional data, we will have hundreds or thousands of measurements on each individual subject, and these become impossible to digest as humans who live in 2 or 3 dimensions. But it's a way of simplifying these hundreds or thousands of measurements down into just a couple dimensions so that we can manage it. And it's also very, very intuitive, the idea of distances. So when it comes to classification of species, we use distances to group together similar species. So we have a bunch of mammals here on the left. You know, we have cat, dog, what's this, a fox and a rabbit. We group together birds and fish. So these are very complex animals. There are all sorts of traits that are unique to each one. We could measure and talk about many things about them, but aggregated among these things, we see that there are similarities that allow us to group them together and have some understanding of them in a simpler way than talking about all of their morphological features or their genetics, which could be millions of nucleotides. These are a standard tool that we use in genomics. Good morning, Angela. The clustered heat map distances are central to this idea of clustering. Whenever you see dendrograms like this, these are representing distances, and in this plot, samples that are similar to each other and features that are similar to each other because they have a short distance summarized over all of the measurements have been clustered together. And we can see, for example, that samples, I don't know, these two that have been grouped together have somewhat similar measurements across many of the genomic features. So distances are the basis of clustering and classification of samples. So fundamental components of machine learning use distances, and really what they are is a mathematical representation of our ideas of similarities and dissimilarities between objects, whether they be samples or genomic features. So as by way of example, in the heat map, when you see 2 columns that you know, they're blue together and then they're red together, that's something that visually to us looks similar. Distances are mathematical representations of that. And there are a lot of different potential representations, and we have to choose ones that correspond to our own ideas of what makes similarity. And as a result, these choices of distances require some thought, and they're subject specific. This is, by the way, a very well-liked tutorial, very old, from 2002, but a good discussion of distances in genomics. They're talking mostly about microarrays here, but it's applicable to all kinds of, most of it is applicable to all kinds of high-dimensional data. Just so that you're aware of terminology, that you will hear discussions of metrics and distances. And a metric is a very specific mathematical definition. It satisfies these 5 properties. So among them are non-negativity, meaning that the distance between any 2 vectors, A and B are vectors here, is always greater than or equal to 0. Symmetry, meaning the distance between A and B is the same as the distance between B and A. The identification mark, the distance between any object in itself must be 0. Then definiteness, related to the identification mark, in that the distance between 2 objects is 0 only if those 2 objects are identical. And finally, the triangle inequality, which just says that the distance from A to B plus the distance from B to C must be equal to or greater than the distance from A to C, which is another formal way of saying that the closest distance between 2 objects is a straight line. And a distance, which is what normally all we require in our applications, is only required to satisfy 1 to 3. Not everything is a distance. We also will use similarity functions and so-called dissimilarity functions, which are even less strict. They only have to satisfy the first 2 of these, and somehow similarity increases as 2 objects become more similar, and a dissimilarity measure decreases as they become more similar. In ecology and microbiome research, a lot of the popular measures that you use to compare communities are actually similarity and dissimilarity measures. I'm going to focus today on the Euclidean distance, which is a metric, because it's one that everyone is familiar with. We learned it in grade school, and it's a very useful one. It is perhaps not the most commonly used distance in genomics, but it is pretty common. So that's all it is. It is the distance in 2 dimensions. We're quite familiar with it. Distance from A to B is just given by the Pythagorean theorem with the horizontal and vertical distances on the short edges of the triangle. By the way, this is also referred to in statistics as the L2 norm. Just so if you see that, you know what they're talking about. Euclidean distance extends readily into high dimensions. So it's not so easy to visualize, but the calculation is essentially the same. And I'm going to demonstrate it here with this data set, the tissue gene expression data set. I'm going to use this for all of the examples today. This is a microarray gene expression data set with 22,000 rows and 189 samples. So these are probe sets corresponding to genes, and there are 189 samples, which come from, what is this, 1, 2, 3, 4, 5, 6, 7 different tissues. A couple from the brain, the cerebellum and hippocampus, colon, endometrium. So we have some distinctive and some similar tissues in here. And in general, what we're going to be interested in doing with distances is identifying similar samples and similar genes. So although our points are not in that nice Cartesian XY plane that we saw before, the idea it will be the same. We have samples I, so one sample is defined by, instead of an XY, but 22,215 points. So we have measurements 1, 2, 3, 4, all the way up to 22 some thousand. And then a feature G is defined by a point in 189 dimensions. Make sense so far? This T stands for transpose, and it's common to, with a vector like this, to put the transpose there just to indicate it as a matrix. And I'll give some examples of what that looks like. The distances basically look like Pythagoras' theorem, except just instead of 2 elements, we have 22,000 elements inside the square root. And we can define that distance between samples summing up to 22,000, or between genes summing to 189. You will see very frequently matrix algebra notation used instead of the summation notation on the previous page. So this is an equivalent way of writing the distance between two samples. So I and J are 2 samples. This is implied to be that vector of length 22,215. So it's saying that we subtract we take the vector for sample I, subtract the vector for sample J, transpose it, and do a matrix multiplication with itself again. And I'll give a little workable example than that, but it works out to be exactly the same as that summation. And then you just take this is a sum of squares, and then you take the square root of it. I won't do too much matrix algebra notation today, not nearly as much as you'll find in the book, but just enough so that you'll get a bit familiar with it. So just to show you what these actually look like, the transpose and the non-transpose, here is a matrix with just the integers 1 to 3 and 1 column. The lowercase t is that transpose that you saw on the previous pages, and all this does is take a 1-column matrix and turn it into a 1-row matrix. So here's the matrix on its own, and here's the transpose of the matrix. So nothing fancy. This is matrix multiplication notation in R. So recall on the previous page we had yi minus yj transpose times yi minus yj. So I'm going to take the transposed matrix and do a matrix multiplication by the untransposed matrix like we saw there. And what happens, it's 1 times 1 plus 2 times 2 plus 3 times 3, which is 1 plus 4 plus 9, 14. So you can see it ends up just being the sum of the products of each element. This yi minus yj transpose times yi minus yj. So far, so good? Great. So let's take an example from our high-dimensional data set I'm just picking out 3 tissues here. We have 2 kidney tissues and 1 colon tissue, and just creating a vector for each of them. So E is the expression data set, the 189 columns by 22,000 rows. So anyone know what this is going to create? What class will be kidney 1? Sorry? All the rows from column 1, that's right. So what class will it be? Like our, we have data frames, we have matrices, vectors, we have.
It's a vector.
It's a vector, yeah. It's a numeric vector. What's its length?
20,000.
Yeah, 22,000 and something. So we're creating 3 vectors here of length 22,000. And I'm just going to take the differences, square them, add up the sums, and take the square root. So this is just the Pythagoras theorem in 22,000 dimensions. And I get a Euclidean distance of 85. And you'll see in the book they give you a few different options for how to calculate this. I'm using a summation method here. They also show you how to calculate it using matrix algebra, which will be faster. And doing the same thing between a kidney and a colon, you see a somewhat greater distance, like we might expect for different tissues. So that's a Euclidean distance calculated in 22,000 dimensions. That's all there is to it. There is also a built-in function for doing this. It's called dist. And dist is a very important function in R that I have used many, many times. It is convenient because it will operate on your entire matrix. So rather than we have 189 samples, so rather than having to write a loop or do something that is going to repeat this calculation for every pair, you can just provide your entire matrix that you want the distances calculated on, and it will do it. So that is one important thing. So here is the expression matrix. I'm subsetting columns 1, 2, and 87. And then note a kind of maybe unexpected aspect. It's something that I forget sometimes, but dist calculates distance between rows of your column, of your matrix. So I have to transpose this to get the distance between samples. If you forget to do that, you probably crash your computer because you're calculating then a 22,000 by 22,000 distance matrix. So this calculates for us 3 distances between these 3 samples. So between this one sample, which I think is our what was it? Our colon sample. We have distances of over 110 to each of the what were they? Liver samples or kidney. And then a smaller distance between these 2 samples, which are the 2 kidney samples. So that's one convenient thing about dist. The other convenient thing is that it has its own class. Its class is called dist. And it is really just a matrix, but the fact that it has been assigned a different class means that R being an object-oriented programming language, there are there are a number of functions or methods which act only on this type of object. And if something is of class dist, it knows how to do certain things like plot a dendrogram, do clustering, various things that you would only do with a distance matrix. It's an important enough function that I thought I would just give an excerpt from its help page. So this is the usage of dist. The first object x is the matrix of which we're going to calculate the distance between all pairs of rows. The second option, when you see equals, it means that the default is Euclidean. And you'll see that it offers a number of different distances that you can use. The only one that I have really used is Euclidean, but that's because I normally work with quantitative continuous data. If you were working with directly with sequence data, so for example, distances between nucleotide sequences would be, you might use a binary distance is another possible choice. And yes, just a reminder, caution, if you did dist directly on E, you create this 22,000 by 22,000 matrix that's got about a, I don't know, 500 million elements or something in it. You probably don't have enough memory in your computer to store that. So just remember that dist operates on rows. I did this a couple of times while preparing this lecture. It didn't actually crash my computer, but it did crash RStudio, and I had to kill RStudio and start over.
What is P?
P. P. Oh, okay, yeah. Let's talk about these other things. So the diagonal, if you set diagonal equals true, it will give you a matrix that has the diagonals. You notice here that the diagonal elements, so between GSM 11814 and itself, is not calculated. So all of the diagonal elements are missing. Any idea why? Why we wouldn't necessarily care about the diagonal elements? They're 0, yeah. So you don't have to have them. Upper equals false, so it's only giving us the lower half of this square matrix. You know why? By default. Symmetric, yeah. If you turn both of these to true, you will see a symmetric matrix with zeros down the diagonal. So its transpose is equal to itself. P equals 2 only matters for one of these other ones, I think Minkowski distance. It's some option to it. Let me see. Actually, let me check that. What is the... Oh, okay, power of Minkowski. So it shouldn't make any difference. Whoops. It shouldn't make any difference for Euclidean distance. Okay. When calculating Euclidean distances, we'll normally use so-called standardized features, where standardization refers to converting them to a z-score by subtracting the mean and dividing by the standard deviation, so that they have 0 mean and a standard deviation of 1 and a variance of 1. And the reason for this, you only do this if it makes sense. It wouldn't always make sense to do this, but for a lot of technologies such as microarrays, such as RNA sequencing, the absolute levels and variability probably has technical biases to it. So the fact that one gene you see more highly expressed all the time than another can be related to GC bias, like there is better amplification of high GC transcripts, and to when on a microarray there are probes that tend to have better complementary binding to high GC transcripts. In some other cases, it might not make sense to do this so. The example of where this would make a big difference is if you have one feature, say a gene that is always highly expressed and varies a little bit around some high level, and another one that is lowly expressed and varies a little bit around the same level. If they go up and down together, they could still have a very large Euclidean distance just because of this shift between them. But after standardizing them, you could find they have a very small Euclidean distance. So it depends on whether, and this is a technology and subject-specific decision. Does this shift between them mean that they're very different? Or are you interested in the fact that they move together in concert? And this is one reason that this 1 minus R Pearson correlation is often used as an alternative to Euclidean distance, because in this case where you have a constant shift and maybe differences in the amount going up and down, as long as they do it together, you'll have a small distance as measured by 1 minus Pearson correlation, which, by the way, is closely related to Euclidean distance after a scaling factor. So if the Euclidean distance is scaled, it turns out to be equivalent to 1 minus Pearson correlation. This 1 minus Pearson correlation is the distance measure really most commonly used in a lot of genomic data analysis. So let's apply these ideas of distance for some real exploratory data analysis by dimension reduction in PCA. Just by way of motivation, because this is a very intuitive example, we're going to simulate a set of weights of twin pairs. So we have in this synthetic data set, we have 100 pairs of twins for a total of 200 people, and we've measured the weight of each of them. This function, MVR norm, will create random normally distributed variables with a specified covariance, or in this case, correlation matrix. And this matrix has ones on the diagonal, meaning that each variable has a correlation of 1 with itself, and they have correlations of 0.95 to each other. So this will create a 2-column matrix that for large enough n, the correlation between those 2 columns will be 0.95, and they will both be random normally distributed with mean 0 and variance 1. So just looking at the dim, we have the dimensions. It's actually done the 2 individuals are in rows, and the 2 variables weights for the 2 people are in rows, and the 100 observations are in columns, which looks a little bit strange when we have only 2 features, but this is the way that matrices in high dimensional data get represented in R, where for every if observations go in columns, features go in rows. It's just this isn't a high dimensional example. So we can check the correlation. Core calculates the Pearson correlation on columns. So I transposed it back and we see they're pretty close. Correlation of 0.943. It's not exactly 0.95 because this isn't too large of a sample, but it's close. So does everyone follow the setup so far? Okay. So here this is a plot on the left of the weights of twin 1 against the weights of twin 2. And we see this high correlation of 0.95. These twins, if one is heavy, the other tends to be heavy, and if one is light, the other tends to be light. And these are centered on 0 with a standard deviation of 1 because they're standardized. And we're just highlighting 2 of them here in red. So this is one twin pair, this is another twin pair. And drawing a straight line between them would be the length of that line would be the Euclidean distance, because this is only a 2-dimensional data set. Now, I'm going to perform a projection here. This is not actually a principal components projection, although it looks, it turns out that this is the principal components projection, but what I did is just calculate the average height and the differences in height. We did this once before already. Do you remember what this plot was called? Anyone remember doing this plot in the exploratory data analysis lecture? Yeah. One of you must remember what it was called. It was for quality control. We used it for, No, QQ plot was doing the normal distribution, so it gave a straight line diagonal like y equals x. This was an MA plot, the rotation of by 45 degrees, that just made it easier to compare the measurements on for two different samples, or for one sample against what I called the median profile, or the median pseudo profile. So this is just rotated, but you'll notice it's also a little bit squashed. So the distance between here is a little bit less. It turns out that there is a scaling also required in order to maintain the distances when you rotate like this. But this is the key aspect here, is that we have rotated into a different coordinate system, where this coordinate system, or the x-axis here, the average height, is your most important variable, and then deviations from that, the differences in height are quite a bit smaller. So you can see here the differences are between -2 and 2 in the average height, but in the differences it's only between -0.8 and 0.8 or something like that. So this one axis, the average height, is capturing most of the information, and that is a form of dimension reduction. If we didn't have room to write down everyone's height, we could just record, for each twin pair, just record what the average is, and we would have most of the, there. There you have class.
Hierarchy for hackathon is shared with this. is booked this room?
I'm pretty sure it's booked for a lecture.
This is reserved for the hackathon.
I think there's an error because we have it reserved for a lecture.
So on the site, it's Morveno, OFEC, and Levicolum for us.
We should. Does someone know Georgia's number? Yeah.
Siamo 20.
Ma neanche ci state, se siete 30. Se siete 30 neanche ci state.
Ma siamo divisi per strade.
Allora
Yes. Ma ti hanno mandato su da giù? Dalla portineria di giù?
No, c'è proprio sul sito, c'è scritto che siamo queste 3 aule.
Non so. Ventotto, 16 minuti.
magari quando c'è un'aula di libera prima la presentazione per le prossime 2 ore. Magari c'è un'aula PCA.
Sì, quindi dobbiamo andare giù.
Sì.
Sì, però no. No, però forse è un dettaglio. Sì, ma I think months ago.
Yeah.
Yeah. So,
Yeah.
Okay.
so,
Okay. Okay.
he seems to be the responsible, I think.
Yeah.
And I think that Angela was going downstairs to check the reception.
Yeah. Okay.
And she was calling Georgia.
Okay. Good. Apologies to anyone watching this later online for the pause. I'm going to just stop here, and I believe I can start again.
Anche a me andresti dico che le gente sarà un conflitto politico, non ci daranno più spazio per le vicine.
Già, bella ciao.
Okay. So the thing to take home from this is that we have achieved a form of dimension reduction where we lose a little bit of information, but not too much, through a rotation. So we've rotated from a 45-degree type of relationship between the heights of the two twins to one where one axis is the average weight and the other one is the difference in heights. And this first axis, the average weight, contains most of the information. So if we were to use only one number, this average height, we would lose a little bit of information, not too much, but we've gone from 2 measurements down to 1. And this is the basic idea of principal components analysis and multidimensional scaling to achieve dimension reduction through an optimal rotation of data onto a different coordinate system. And here I've actually just plotted the pairwise distances between all pairs of these 100 individual, and looking at the distances in 2 dimensions compared to the distance in 1 dimension. And for the heavier twin pairs, there is almost no loss of information if you only look at the average. Okay, I just am going to pause for another moment. Okay, so we only are really losing any noticeable amount we're only noticing any change in the distance for the very, very lightweight twins, which makes sense because the distances the way we've constructed it, the differences between the two are pretty constant, regardless of whether the twins weigh 20 kilograms or 60 kilograms. The differences between them are the same, which is not totally realistic. It's a synthetic data set, but that's why we see this occurring down in the bottom left corner. But the important thing is that by in doing a dimension reduction from 2 to 1, we really haven't lost very much in terms of pairwise distances, and this would allow us to plot these distances to look for say similar sets of twins based on their average height using one dimension instead of using two, which isn't a big deal here, but when we have 22,000 dimensions, it is a big deal. Singular value decomposition, SVD, is a mathematical approach for performing rotations and decompositions such as this in larger data sets, and for determining the rotations when it's not just a case of doing something like average versus difference. I like this graphical representation of it very much. If you're not too familiar with matrix algebra, this sort of helps to demystify what it is. So this Y matrix, and I'll go over these again, but this is a matrix of our actual observations. So this, in our example, is the 189 columns by the 22,000 rows. And what's happening is it's being broken down into 3 different matrices. The first is another matrix of the same dimensions times a diagonal matrix, meaning it's only non-zero on the diagonal terms.
C'è RTTR che
Era in inglese perché stavo in inglese.
scusatemi,
We need a sign outside that says no hackathon. Or we could invite them in to do some principal components analysis in a little bit. Yeah. Okay, so I'm going to discuss each of these in detail because the interpretation of these is quite important for principal components analysis. But for now, know that we're breaking this 22,000 by 189 matrix into another matrix of equal size and 2 diagonal matrices. Two square matrices with rows and columns equal to our numbers of samples, one of which is diagonal. And this is specifically for the high dimensional cases where we have more rows than columns. It's a little bit different, the decomposition in non-high dimensional cases, but I'm only gonna give the formulation here for high dimensional data. Here it is, just with each of these defined for you. Again, Y are our actual experimental measurements, which are being decomposed into these 3 matrices. U being again M by N, and this is an orthogonal matrix. Orthogonal means that these columns and the rows are in 3 dimensions. We would think of it as being at right angles. So if this were drawn on the table, it would look like that, a right angle, not an acute or obtuse angle. The definition is a little bit different when we're talking about longer vectors. It has to do with the inner product or dot product, which I'll define in a bit. But you can also just think of orthogonal as meaning that they're independent. So these columns are not correlated to each other. They're orthogonal. D is this n by n, so number of samples by number of samples diagonal matrix. It only has elements along the diagonal. And these diagonal elements are the eigenvalues, to use language from matrix algebra. And then V is another orthogonal matrix, this one n by n, so 189 by 189 in our example. And these are the so-called eigenvectors, or when we get into the language of principal components analysis, the loadings. I'm going to actually go ahead and calculate this singular value decomposition. And I'm first going to create a standardized data set where here I'm only going to subtract the mean from each one. And I just did this for your interest in 2 different ways. The first way, a slow way, I'm using this apply function, which in R is an implicit loop. What apply does is take a function and apply it to all of the rows or all of the columns of a matrix. So apply, its first argument is our matrix. The 1 means to apply the function that I give it to every row. And the function will just be to subtract the mean. So for every row, I'm subtracting the mean, row by row. And then I have to transpose it to get it back in the same order that I had. So this is kind of the DIY, or the do-it-yourself manual way, and it's also fairly slow because it's a loop. It's not too slow because this is not a huge data set, but as is often the case in R, if there is a built-in so-called vectorized function that takes your entire matrix or your entire vector and performs the computation using a subroutine that is probably written in C or Fortran, you can do the same thing much faster. And there's a scale function that is specifically for doing this, for scaling and or centering the columns of a matrix. So the reason I have to transpose is because I'm scaling, or I'm centering the row. I want to center the rows, not the columns. So I transpose it, do the centering, and then transpose again. And all of this occurs about 10 times faster than if I use this implicit looping function. R has an enormous vocabulary. It has a vocabulary of, I don't know, thousands of functions. And as you learn some of these specialized functions for doing something specific that you want, in general, it will be much more efficient than using the generic way, using something like apply and mean that it have many other uses. Speaking of very specific functions, although this could be performed with matrix algebra steps, which the book shows you how to do, there's also a built-in function that performs singular value decomposition. And I just provided this standardized data set, and it does this quite fast. it returns to us a list object with 3 elements, D, U, and V, which conveniently correspond exactly to DU and V in the terminology that we're using. So we can take a look at it. The U, these loadings, it's that M by N matrix. Sorry, I think I reversed this. These should be the scores. These should be the loadings that M by N. Yeah, these, sorry, this should say scores. I'll correct that later. By the way, not everyone agrees on this notation, which might have been why I did it. When I first learned, I used to call these the loadings, and then I relearned it because I think, well, the R functions refer to these as scores, and the book also refers, the book doesn't mention scores, but it definitely refers to these as loadings. I think it's more standard. But anyways, the U matrix are 22,000 by 189. We've got the D, our N by N diagonal matrix, which it just represents as a vector, so it doesn't have to bother storing all of the zeros on the diagonals. It just stores the actual diagonal elements as a vector. And then the S, or the V matrix, 189 by 189. So this SVD function has given us these 3 elements. There's another function, which is the one that I actually prefer to use. It's a little bit more convenient and has some additional options built into it, like scaling and centering, so that you don't have to do those in a separate step, called princomp, but it will give you the same results as SVD. It is an SVD. And I just show this by plotting the eigenvalues from each of them. They're the same. And I'll tell you in a little bit why I'm calling this percent variance explained, but that is the way that the eigenvalues are often described in an intuitive way. Okay, so first this U matrix, the M by N matrix of scores. These are important as their interpretation is they're the matrix that relates the original variables to the principal component variables. It provides it's a rotation and its values give you the relative importance of each of your variables to each principal component. So the N, the columns will correspond to principal components, and the N correspond to your original variables. And you can think of one of these columns as being the weight in a weighted average of telling you how you convert an original, your original variables into principal components. So a very large entry in column 1 will tell you that this variable is important to principal component 1. And a value near 0 will say that this variable is not contributing to principal component 1. So to give you an example of that, I'm taking, you see that this is a reason why I like the princomp function instead of the one that the book uses. The book uses a different one, but its terminology is a little bit different. They do essentially the same thing. But I'm taking the scores first, the M by N matrix, the first column. So this will be the scores for the first principal component. And I'm just plotting them. So this is just an unsorted line plot. There are 22,215 data points here. This is the score for each one of the variables for principal component 1. I drew lines in a couple of arbitrary places to tell you that I'm going to pick out all of the original features, so all of the original genes that have a score greater than what is it, 25 or less than -35. So these are the most important variables for principal component 1. And all I'm going to do is take the original gene expression matrix and select out the genes that have these extreme scores for PC1. Does this make sense?
Is there a meaning in the negative score?
Yes, there is a meaning in the negatives. Well, so there's a meaning that these scores have the opposite sign, or these genes have the opposite sign in the scores as these ones. It turns out that you can switch all of the scores and then just switch the corresponding eigenvalue. So the sign is arbitrary, but the fact that the sign is opposite is important. And I made a heat map on the next page to show you why.
But I'm lost.
Okay. So on this here, I'm looking at each of the 3 decomposed elements, and I want to explain what each of them means and how we interpret them. And we're starting with this first matrix, which has the same dimensions as our original matrix. And these are called the scores. And what I want to describe is that these tell you the contribution of each of your original variables, let's say genes, to each of your principal components. So if we talk about principal component 1, it's, it may be a useful thing. Maybe we find it is associated with the tissue type and that, but what is principal component 1? How do we interpret it? We can interpret it using these scores, this U matrix. So principal component 1 will be defined by the first column of this matrix, and the genes that have a very large value are contributing a lot to that principal component. Literally, what it works out to be is so without doing too much matrix algebra, if we multiply the left side of both of these equations by U transpose, because U is orthogonal, U transpose U is an identity matrix. So we can put the U transpose on the left here. And when we convert, and that would allow us to convert our original gene expression matrix into the principal components matrix. And the way that multiplication would work is we would multiply our expression value. So let's say we're working on patient 1. We want to know what the first principal component is for patient 1. We have a vector of 22,000 gene expression values, and we have a vector of 22,000 loadings. We would multiply each of those gene expression values by each of the loadings to get the principal component. So it matters not only what the gene expression values are, but what the score is for that gene. And the gene that has a high score is going to contribute more to the principal component. Good. Thank you for asking. I want to make sure that the meaning of each of these is very clear to you. So if you're at all unsure, we have time. Just ask, and I'll find another way to explain it. So does this make sense, what I'm plotting here? Can you understand the code here, what I'm doing? This plot, this is just plot on a vector, and all it does is it puts the index there's no hackathon. Okay. So on the x-axis is just the position. This is the first row. This is the 22,000th row. And these are all of the scores. These are the most important genes with a positive contribution to PC1. And these are the most important genes with a negative contribution to PC1. So if we were calculating what is the value of PC1, we would take the expression value of this gene and multiply it by 25, and the expression value of this gene and multiply it by negative 41. So their importance is really magnified in PC1 relative to all of these ones in the center that get multiplied by something smaller. So I'm just going to pick out these genes. So which of these scores is less than minus 35 or greater than 25, and then make a heat map. And you can see we have two blocks of highly correlated genes here, and they are anti-correlated to each other. You know why they're anti-correlated? Did you expect that? First of all, did you expect to see such highly correlated genes here? Obviously, if we just picked, we have about 50 genes here. If we picked 50 random genes, they would not be nearly so highly correlated. Can someone explain to me why these genes are so highly correlated? Or maybe not why they're correlated, why we picked such highly correlated genes? This is a very key component to understanding principal components analysis, this correlation. It's not an accident that we picked a bunch of extremely highly correlated genes here.
Selected them.
Mm-hmm. We selected them so these principal components are ordered. They're ordered by their eigenvalues, which I'll describe in the next slides. But they're ordered in descending size of the eigenvalues, which turns out to be descending size of their variance on the rotated scale. So you remember with the twin heights, after the rotation, one direction had quite a bit of variability and the other one had less variability. It is the same thing here, and we've rotated it so on this first, for this first principal component, there is a high amount of variability. And the way this happens is by rotating into the direction with the greatest amount of correlation between features on the original scale. So if you have correlated genes or weights of twin pairs, it allows you a large amount of dimension reduction. So you can see that there's actually not a lot of independent information in these 10 or so genes. If you were to just report the average expression of them, you would have a fairly close idea of what the expression of any one of these genes is. And this is how we're achieving dimension reduction by the principal components analysis. So so the construction of the principal components analysis is to create a PC1 which in which the contribution of it according to these scores will come from highly correlated genes who essentially we don't need to represent the measurements for each of them. We can use this one set of measurements to capture values for these many genes at once. So the reason that we have these two blocks of anti-correlated measurements is because we have some with the positive scores and some with the negative scores. Does anyone want to guess which is which? Which of these are the positive scores and which are the negative scores? We could figure it out, but I can make a pretty good guess here. I'll give you a hint. Notice that the values of the negative scores are quite a bit larger. They're -40, and they're only about scores of around 25 to 30 for the positive ones. So these negative ones have a much stronger contribution to PC1. Which of these blocks looks like it should contribute more to PC1, this one variable that helps define a pattern of a whole bunch of genes? So which ones are these, positive or negative scores?
Negative.
No, so the rows are genes. This horizontal row, this is a bunch of genes. So one gene has one score, and it's either positive or negative. And all of these genes, because they have the same pattern, they will all have positive scores or all have negative scores. So does this much make sense to everyone? What do the rows represent? What do the columns represent?
The rows are the genes that we selected.
Yeah, that's right. The rows are the genes. Yeah. So the rows are the genes. We have these two blocks that are highly correlated within a block and anti across the two blocks. We can recognize those together. These are going to be correlated to each other and anti-correlated to these genes. Because if we're going to, in PC1, get rid of all of these measurements and replace them with a single value that approximates expression of all of these genes without too much loss of information. So obviously, if you added these together, you'd be losing something. You'd get something just coming out around 0 that's not going to tell you whether a sample has higher expression like these or lower expression like these. So you're going to have to obviously take a difference between expression values for this block of gene and subtract expression values from this block. So as to the question of which of these are the positive, which of these are the negative scores. So I noted here that the negative scores are making a stronger contribution. I noticed that these have much better correlation. They're much more tightly correlated than these, which tells me that these should be making the stronger contribution to PC1. We're losing less information by representing all of these as a single number. So I'll bet you that these are the ones with the negative scores on the previous graph. These are a little bit less coherent, so we wouldn't want to count them as much in this summary. Any questions about this? Are the scores making sense to everyone? This weighted combination of the original genes. Oh shoot. Okay, I think it's okay. Okay. Thank you. Feel sorry for the people tuning in online today. They'll wonder what all is happening. Just so you don't wonder too much, I spilled some water on my computer and I'm drying it up now. Okay. Okay, the second element in that decomposition, the one in the middle, the D, the diagonal matrix of eigenvalues. So these will be ordered in descending order, and because both of the other elements, the U and the V, are orthogonal, they are also unit length. So if that middle term, if it were all ones, then in our rotated decomposed space, every variable would have the same variance. All of the variance comes from this diagonal D matrix. And you can think of it as a scaling factor for standard deviations. So a principal component with an eigenvalue of 1, that variable will have a standard deviation of 1 in the rotated space. A value of 2, a standard deviation of 2. So it tells you about the variability of your genes in this decomposed space, or sorry, the variability of your variables in the decomposed space. And we can just plot it directly as that. So I'm going to square it to turn these standard deviations into variances and just plot so this is a vector of length n, 189, plot the square of them, so the variance of each of these components, divided by the total, multiplied by 100 to turn it into a percentage. And this produces what we call a scree plot, which is the percentage of the total amount of variance added across all of the decomposed variables for each one of them. And you see how they're ordered. The first one has the highest standard deviation and variance, and then they're monotonically decreasing. And once we get up to about 20 here, we have essentially 0 variance, or 0% of the variance for each of them. So in this actual gene expression data set, even though we have 22,000 measurements, we can represent almost all of the the variability in them using only about 25 variables or principal components, which is pretty incredible, right? We could reduce these 22,000 variables down to about 25, and from them, by performing this multiplication of the three matrices, you could reconstruct all of those gene expression measurements quite accurately. It's a huge, huge dimension reduction, and it's a reflection of the fact that these transcriptomes are quite highly correlated. There are not too many truly independent genes among them. Pretty neat, right? I think it's neat. This doesn't always happen, by the way. You can have a scree plot where, you know, if you have a lot of really independent variables, where it takes a long ways to level out. This is the other way scree plots, or one other way that scree plots can be represented as the cumulative percentage of the variance. And all I did was put this function cum-sum for cumulative sum around the first element, so that it rather than plotting the actual variance, it adds all preceding elements to it, so that by the end we're at 100%. And you can see we get up to 90% in about 25 variables. Okay, finally, this final element of the matrix. So as you see, each of these decomposed matrices, they're really important, not just for the mathematics, but for how you interpret principal components analysis decomposition. This final matrix V are for the so-called loadings. These are the actual data points in the decomposed principal component space. And they're the ones that, when you see principal components plots like this, these are the loadings. This is just the first two columns of that V matrix. And literally all I did was take the loadings element of this print comp output and plot the first two columns. And then I defined changed the colors and the data point types according to tissue. So in this enormous dimension reduction from 22,000 variables down to 2, we can actually see some pretty interpretable differences here. So we've got one block quite distinct here. These are the liver samples. They look like this. So they have consistently different expression patterns than all of the other tissue types. Looking over here, there are two that look not too distinct from one another. These pink X's, hippocampus, are kind of enclosed by the green dots, cerebellum. I don't know too much human physiology, but I'm pretty sure that both of those are parts of the brain. So I guess it makes sense that they're appearing together. They have not entirely different expression patterns. And then over here, another group of kind of overlapping samples, and these are colon, kind of distinct from the kidney, but then not so distinct are the placenta and endometrium. So pretty neat, right? We took something. If you look at a 189 by 22,000 matrix, all it is a whole bunch of numbers that you can't readily interpret. We've reduced all of those measurements down to just 2 measurements per sample and plotted them and seen that that this plot is interpretable. So I thought today we would do a couple of labs, just shorter labs, kind of interspersed with the lecture. Do a couple of things on PCA, and I'm going to continue with the multidimensional scaling, of which there isn't too much to do, and then you'll have a take-home lab as well. So I'm just going to pause the Hangout again.